Continuation of previous video about transcriptional control. So next model, Britton-Davidson gene battery hypothesis. Britton and Davidson in 1969 proposed a novel mechanism for transcriptional regulation in eukaryotes. According to him, hormones and other regulatory molecules can control eukaryotic gene expression through the mechanism of a gene battery. And Britton and Davidson hold that eukaryotic genes which govern a metabolic pathway are not linked but located in different chromosomes. Still then they are coordinately regulated, just as in the case of prokaryotic operons. Britton and Davidson visualize 4 kinds of hypothetical genes, namely sensor genes, integrator genes, receptor genes, and producer genes. Sensor genes, integrator genes, receptor genes, and producer genes. These collectively constitute the so-called gene battery. Sensor genes are detectors sensitive to the state of cell and its environment. Producer genes are the structural genes coding for proteins. Integrator genes are positive regulators which mediate the expression of 2 or more contiguous producer or structural genes. According to the gene battery hypothesis, sensor gene is stimulated by regulator molecules such as enzymes, hormones, metabolites, etc. The stimulated sensor gene induces the integrator gene to undergo transcription to produce an activator RNA. Receptor gene detects this RNA and gets activated by it. The activated receptor gene induces the producer gene to undergo transcription and translation. This results in gene expression. Sensor gene induce integrator gene, then lead to transcription. Activator RNA, then receptor activates receptor gene that induces producer gene, then transcription, mRNA translation, and gene product, gene expression will be happening. So that is something which is explained by gene battery model of Britton and Davidson. Then next, transcriptional control by hormones and cytokines. Hormones and cytokines are the chemical agents which influence their target cells by activating the transcription of specific genes. Cytokines and proteins whose target cells are mostly blood cells. They modulate gene expression in target cells in different ways. Steroid hormones are lipophilic, so they directly penetrate the plasma membrane of the target cells and bind with specific transcription factors or interior receptors called steroid hormone receptors. Steroid hormone receptors. The receptor molecule is in an inactive state since it remains complexed with an inhibiting protein. Its binding with the hormone activates it by removing this protein. The activated hormone receptor complex now serves as a gene inducer. It selectively activates the transcription of a specific gene by binding with its promoter or enhancer. Peptide hormones and cytokines regulate transcription in a different way. They are large sized and lipophobic, and hence they cannot directly penetrate the plasma membrane. So they bind with specific surface receptors or proteins of target cells. The hormone receptor complexing with the plasma membrane triggers gene activation by a process called signal transduction. It involves the participation of CAMP and the sequential activation of a series of proteins by phosphorylating them. This leads to the binding of transcription factors to the promoters of their target genes, stimulating transcription. Then post-transcriptional regulation in eukaryotes, regulation by mRNA processing. Post-transcriptional regulation of gene expression in eukaryotes involves the post-transcriptional processing of the primary mRNA. Eukaryotic genes are mostly split genes with alternating exons and introns. So the primary mRNA is transcribed to them also will contain exons and introns. This primary mRNA is subjected to a precise process of chemical tailoring called gene splicing. During it, introns are sliced away by ribonucleases and the remaining exons are spliced together by RNA ligases. This forms a secondary mRNA which contains only exons. The secondary mRNA undergoes genetic translation to accomplish gene expression. This post-transcriptional gene splicing regulates gene expression in such a way that only the exons are phenotypically expressed, discarding the introns. In mammals, there is a special type of gene splicing called alternative mRNA splicing. It involves different types of splicing in different types of cells and in different stages of development. This differential splicing or alternative mRNA processing will produce several types of mature mRNAs which differ from each other only slightly. This in turn will result in the production of different varieties of the same protein, and this will generate diversity of gene expression. This protein differ from each other very slightly in their amino acid sequences and functional properties. And alternative mRNA splicing, some specific splicing sites are joined together while the others are bypassed according to the cell type and the developmental stage. Then translational regulation of gene expression. Translational control of gene expression involves the regulation of the rate of genetic translation through a process called differential translation. Differential translation. In this case, translation occurs at different rates in different cell types or in different stages of development. The molecular mechanism involved in this process is not definitely understood. A typical example of differential translation is furnished by unfertilized or fertilized ova. Both have a large stage of storage of mRNA. Still, the rate of translation is very low in unfertilized ova and very high in fertilized ova. Then post-translational control of gene expression. This is accomplished by two mechanisms, namely control of the cellular levels of proteins through their degradation, feedback control of protein synthesis. The cellular concentration of all proteins remains fairly constant. At the same time, some proteins are produced in large amounts through continuous translation and some others in small amounts through discontinuous translation. The proteins produced by continuous translation are rather unstable and short-living. They undergo degradation at high rates and this regulates their cellular level. On the other hand, the proteins produced by discontinuous translation are relatively more stable and long-living. They undergo degradation at very low rates. In eukaryotes, protein degradation occurs by activity of proteolytic complexes, multi-catalytic endopeptide complexes called proteasomes. In feedback regulation, the cellular concentration of some proteins, especially enzymes, regulates their own synthesis. Their concentration above the normal threshold level inhibits their further production. This is negative feedback. Similarly, their concentration below the normal level stimulates their further production. This is positive feedback. Next is about regulatory RNAs, which will be explained with the help of next video. Thank you for listening.